membrane-bound structures called eukaryotic cells differ from prokaryotic cells in that they contain a nucleus and other membrane-bound structures called organelles. Eukaryotic cells can be a plant, animal, or fungi. Many single-celled organisms like protozoa and all multicellular organisms are eukaryotes. The endoplasmic reticulum is a contiguous complex network of interconnected tubes and membrane sacs that extend through the cell's cytoplasm. This complex makes up roughly 1/2 of the membranous tissue of the cell. There are two types of endoplasmic reticulum, the rough and the smooth. The rough endoplasmic reticulum, or rough ER, gets its name from the fact that its surfaces are studded with ribosomes. The membrane of the rough ER is contiguous with the nuclear membrane as well as that of the smooth ER. The rough ER's primary functions are to manufacture and then fold proteins into their final useful configurations. The proteins it produces are then either secreted from the cell, transported to locations within the cell where they are needed, or sent on to the Golgi apparatus for further processing. The cytoplasm is a semi-fluid, gelatin-like substance that surrounds the organelles inside the cell. It acts to cushion, protect, and support the organelles, and fills all the empty space with the cell membrane not occupied by organelles or other structures. Although the cytoplasm is made up mostly of water, its structure and properties are not fully understood. There are multiple levels of organization within the cytoplasm, including concentration gradients of ions and complexes of metabolic enzymes. The concentrations of ions such as sodium and potassium are different in the cytoplasm than in the extracellular fluid. These differences in ion levels are important in processes such as osmoregulation and cell signaling. Nuclear pores are hollow protein structures that penetrate the nuclear membrane and act as doorways between the nucleus and the cytoplasm. They allow small molecules and ions to freely pass into or out of the nucleus, but also may allow larger proteins to enter the nucleus from the cytoplasm if the proteins have special sequences, or tags, that indicate they belong in the nucleus. In the same way as proteins are allowed to enter, RNA transcribed in the nucleus and proteins destined to enter the cytoplasm have export sequences that tag them for release through the nuclear pores. Mitochondria are the power plants of the cell. They convert sugar into ATP and enzymes related to cell metabolism. Mitochondria are unusual in that they are surrounded by two membranes and contain their own independent genome. They also can divide independently of the cell in which they reside, meaning that mitochondrial replication is not tied to cell division. The number of mitochondria found in a cell varies by the type of cell. The more energy a cell needs, the more mitochondria it will have. Many cells contain only one mitochondria, while others may contain as many as several thousand. Mitochondria are found in both plant and animal cells. Inside each human cell, there is approximately 6 feet of DNA existing as 46 chromatin strands. Normally, this DNA is dispersed in a somewhat loose configuration. However, when the cell is preparing to divide, each strand of DNA is condensed by a factor of 10,000 times into a structure called a chromosome. This folding and repackaging serves to facilitate the process of cell division. When the cell divides, the chromosomes separate, providing a complete copy of the generic information to each cell.